Okay, thank you for waiting, and thank you everybody for joining this webinar. So once again, good morning, good afternoon, and or good evening to you wherever you are listening and watching. My name is Harold Bornfleet, and I am the product manager for BESA Research. Today I'm going to speak about the latest feature in the BESA Research software. As you maybe know, we released version 6.1 in November of last year, and since then we have added 2 minor releases for small improvements, with a third one being imminent. So in the coming hour, I would like to walk you through the major new features of this new release of BESA Research. The menu for today is, first I'll give you a short introduction of the main new features, which comprise cortical imaging methods, age-appropriate template models for source modeling in children and young adult data, and source montages for resting state network analysis. We will then see a hands-on demonstration of these features, and after that, there will be time for questions. So please, throughout the presentation, feel free to send in your questions using the chat. My colleague Avinash Pant, who is with me here at BESA headquarters, will pick up all your contributions to the discussion, and we'll then try to answer them at the end of the session. So, what's new? First of all, let's have a look at the new cortical imaging methods. These are the cortical Loretta and cortical Clara. Minimum norm is also available. We can proudly say that the computation in BESA Research is performed on the actual individual or on the standard cortical surface, and it's not just a simple projection or a cutout of source space. So the results can be shown on the reconstructed gray-white matter boundary or on the inflated cortex. And here we can now benefit from the automated cortex inflation tool that was introduced in BESA MRI version 2.0, which is actually a free upgrade for those who already use BESA MRI. If cortex inflation was performed on an MRI dataset, it is automatically available in the BESA source analysis module. We can also seed dipole solutions from the results of the cortical imaging. To do this, BESA finds the local maxima of activity, and the user can place dipoles at the positions of these maxima. The advantage of this is that you can immediately see the time course of activation, and it can be quantified, for example, in statistical analysis. For the new imaging methods, the most difficult task was to port the Laplacian operator from the 3D space to 2-dimensional space. After all, we wanted to implement a surface imaging method. So the Laplacian operator is a crucial part of the Loretta solution. Without the Laplacian, it is just another minimum norm approach. But the Laplacian is only well defined in a structured grid. So for a surface mesh, we need something more adaptable to local variations in the surface structure. We worked with the University of Sofia to evaluate the performance of several approaches to realizing the Laplacian on a 2-dimensional surface mesh. The best Laplacian seems to be the unweighted graph Laplacian, which is the most robust in the tests that were performed, and you can see that on the left here. We also added more solutions. The next one is the weighted graph Laplacian, and finally the most complex one implemented was the geometric Laplacian with area weights, where we're not just looking at the neighboring mesh nodes, but also at the areas spanned by the neighboring triangles and the angles that these triangles open up. So there's actually a fourth implementation, but in the study this one performed worst, and it was removed from the options available. We also have a publication in preparation by Jordanov et al., which will be published soon. And so the cortical Loretta method gives you a wide signal distribution of the cortex, and we are often interested in a focal result. So to achieve that, we implemented the cortical Clara. And cortical Clara is a cortical Loretta that it uses as an input, and then it proceeds iteratively. And in each iteration, the source space is reduced. We only keep the 90% of nodes with the highest signal. We can also weight the image of the current iteration with the result from the previous iteration. And this will gradually focus the result to the underlying generators. A first publication for this, using this, was out this fall in the journal Seizure, which compared cortical clara and other imaging methods in several epilepsy cases. Yeah, so when you have very focal results, it's possible that these are no longer visible because they disappear in a fissure. In that case, or in many other situations as well, it can help to have an inflation image of the cortex. This keeps the information about the gyration, but it also gives you the chance to see the complete hemisphere at one glance. The inflation is now available in BESA MRI. It required some substantial rework of the automatic segmentation in order to fix the famous T-handle problem. The T-handle problem is the problem that if you have a teacup with a handle in it, it's impossible to morph its topology on an object that does not have a hole in it. And we don't want holes in our reconstructed brain hemispheres. Yeah, anyway, we fixed that, and it's now just another checkbox in the automated segmentation workflow of the brain. The nice thing is that you don't have to think about integrating the inflated surface with BESA source analysis. If it was computed, it is automatically available after co-registration. And if you don't have BESA MRI, you can still use the inflated brains of our template models. Yeah, that brings me to the next topic. So another new feature in BESA Research 6.1 are the age-appropriate template models. The data for these models were kindly provided by John Richards from University of South Carolina. And if you use these models, we would kindly ask you to cite the papers that are shown at the bottom of this slide. You can find this information also in the BESA source analysis help file if you go to the chapter on head models and then to the subchapter on age-appropriate template models. The models have been computed for children with age ranges of 2 years gaps. So they start at infant age and then move in 2-year steps up to approximately 20 to 24 years of age. The nonlinear co-registration of the brains that was used preserved the internal structures. So you can easily recognize the gyri in the model anatomy. For children of 12 years or older, we also have the inflated models available. For younger ones, we don't yet, but we have a PhD student who right now works on precisely the automated segmentation of young children's MRIs. So please bear with us for some great results there as well. And the head models that we here have now give you the chance to obtain accurate source modeling results for younger children too. So the conductivities in the head shape can be quite different for these. It's important to have these models available if you don't have individual MRI. Yeah, and finally, we have also launched source montages for resting state networks. In a literature review, we figured out that there's not only the default mode network at play, but there are 4 different networks that seem to be important for various brain functions and which were identified by fMRI and PET studies. So we found the underlying sources and created source montages for them. We have the default mode network, the dorsal attention network, the frontoparietal task control network, and the ventral attention network. You can directly select the source montage in the BESA Research review window. It then contains the sources of interest and also some noise sources which are used to model unrelated brain activity. If we didn't have these, unrelated activity might spread to our sources of interest. For MEG data, you can load the corresponding source model in the source analysis package, and then you can apply it to the data as a source montage. So how do you apply these for your analysis? Well, for an example case, after cleaning or rejecting artifacts, you can select the montage and then perform time frequency analysis on the montage. And then you have several options. Either export data for statistical analysis on group differences, or you can run a beamformer on a subrange in the time frequency data and then find a brain voxel with high amplitude. For this voxel, you can then run the DICS algorithm to find the coherent activity in other brain regions. Or you can just run the coherence analysis on the source montage directly too. At any stage of these, you may export the results for further statistical analysis. Yeah, did I mention yet? We will have another webinar next week where we will focus on statistical analysis. Right now I have mentioned. And yeah, this actually concludes our introduction. And now I'd like you to show how all this looks in practice. So let's have a look at the BESA Research application. And so I'm going to start with an MEG example. And in this experiment, the subject watched a screen and there was a static random field of dots, or there was a static, the random field of dots that moved towards the subject. So we had two conditions, one that's called motion and one that's called static. We can see these two average conditions here, left and right, and I can get a map of my data. So I can see in the static condition that I have a typical dipolar pattern over the occipital part of the head. And I can look at a sequence of these maps by clicking on one of the heads. And I can also look at the same map for the motion condition. And in the motion condition, I can see the same initial activity. And if I change the gap between maps, I can see that later on the activity seems to spread to other regions. And if I go back to the static, then the activity spread there's also something happening, but not quite as strongly pronounced. So let's analyze this in more detail. We do actually have the individual MRI for the subject. So I'm first going to co-register this data set with the reconstructed brain. So let me go to File, MRI co-registration. And when I do this, then BESA asks me to save the file that contains the head surface points. So I will save this under flow field test3.sfh. And the next thing is that BESA will automatically start the BESA MRI software. And I can now start a co-registration workflow in the BESA MRI software. And I can get through my workflow by interacting with the workflow on the right-hand side of the screen. And in the center of the screen, I will get shown any updates or results that have been achieved. So I can actually go to the next step simply by pressing the next button or the space key on my keyboard. The steps I want to do for co-registration are simply to co-register. I'm not going to generate MEG FEM because it simply will take longer than we have time in this webinar. And I can actually get some nice results also with the spherical head model for MEG. So next step is to select the input project. And the input project is now here already selected. I can simply open this. And I can see that the next step here is to define the fiducials on the reconstructed head surface. They have previously been defined for another co-registration, so I only need to do that once for a subject. And if I'm happy with that result, I can simply proceed, pressing the spacebar again. And now go to the next step to select the co-registration file. This co-registration file is the one that I just saved from BESA Research. So I find it here and open it. And BESA MRI then performs an initial fit that's only based on the fiducial positions. And that fit is not yet very accurate. But if I go and run the advanced surface point fit, then I can see that my fiducials and other points are now nicely matched to the head surface. The pink spheres here denote the fiducials that were digitized, whereas the blue ones are the ones that I identified on the head. I've also got some additional green spheres here, which are MEG coils used for co-registration with the MEG helmet, and some additional skin points in gray. The overall result shows me that the mean distance error is 0.92 millimeters, so that's a very good result. And I can just finish this co-registration by clicking the finish, save it for later, and go back to BESA Research. In BESA Research, I can see that the fields for the MRI and the surfaces have been filled in, and I can leave this box with OK. And now I want to explore the source imaging for the motion condition. I can drag over a data block and then right-click and select source analysis here. And then I get an additional dialog that allows me to refine my selection of data interval. And I will now choose from data interval from -100 to 400 milliseconds. And filters I should set for between 2 and 40 hertz. This looks like a good choice. And start the source analysis. Right, and now I'm also going to look at the static condition. So I can see if I go back and minimize this window and drag over the static condition and again right-click, I have an additional menu entry here at the bottom where I have exactly the settings that I used for the other condition, and I can send this data block up to source analysis 2. And I can see now the global field power here in blue, where I see one peak for the static condition, and if I change to my motion condition by using these arrows at the left, I can see there are 2 peaks appearing. So obviously the second peak seems to be unique to the motion condition. Right, so now let's do some cortical imaging. So let me double click into the first peak and select the imaging method Cortical Loretta, which is already pre-selected, which then starts computation of the cortical imaging. When I start the cortical imaging for the first time, there will be a delay in computing the result because BESA now needs to compute the interpolation rules on the individual cortex mesh where the result is displayed. And this takes approximately 30 seconds on a standard computer. May take slightly longer now that I'm broadcasting my screen. So yeah, meanwhile, I can just mention that in the source analysis window, the left-hand side here shows you the measured data, so the channels that are recorded, in this case MEG channels, 121 channels, and the butterfly plot of the whole signal. Okay, and now the computation is complete. I can see the co-registered brain surface and the cortical Loretta result. And if I rotate this brain, I can see that the main activity is at the very occipital tip of the occipital lobe. And I can now check what's happening on the second peak. So this is much faster. I can see how the activity is now lateralized strongly to the right, and I can use the cursor keys to go back to my first peak and see how the activity changes between initial primary activation and then later. Okay, but this is cortical Loretta, so it does lead to a widespread activation in the reconstruction by definition. So let's try the cortical Clara for comparison. So I select the cortical Clara from the arrow menu here. And now, yeah, that takes a few iterations till it gets to the result. And finally, I get my result. So now the activity is much more focused on one spot, so it's still the occipital cortex that's activated. I can try and click the inflation button to see where the peak is. So I click the inflation button here, and it'll now do the same thing again, calculate the interpolation of the result on the inflation cortex. So it'll take a few seconds again to get through that. And then we get the results presented on the inflated cortex, which should allow us to see everything that's going on at one glance. You can see the toolbar buttons here in the 3D window. So the leftmost toolbar button is the one where I switch between a slicing anatomy view and a 3D cortical surface view. And then I've got a set of toolbars that allow me to navigate the scene. And this new set of toolbar buttons here with the inflation button is the one where I can toggle between inflated and not inflated cortex, and the next two buttons I'll explain in a minute. So here we go. This is the inflation finished, and I can see the peak is now very much focused on just the occipital cortex. There's a very small activation here already lateralized on the right, and let's see on the second peak. So computing cortical clara on the second peak. A few iterations. And then I can see this lateralized activity on the right, but also some on the left cortical surface. So obviously something happening in a later point in time there. And I can now try and see dipoles from these local maxima that were found in the computation of the cortical clara. So let me go back to the individual cortex without inflation. It just needs to recompute this result for the uninflated cortex. And I now have these two buttons here available, and the first one allows me to look for local maxima in this activity. So if I click on this button, I can see that there's now 6 local maxima, and the first one is automatically selected, so I can see this little white sphere appearing, and I can now see the source at exactly the position of that local maximum using this second toolbar button here. So I can see that it sees the source and immediately fits the orientation at that position for this source. And I can then toggle to the next maximum. So maximum 2 is just beside it, and maximum number 3 is now actually on the other hemisphere. So again, I could see the sort from here. Just simply press this button here. And I can have a look at the other maxima, and as far as I remember is that, there's something here in the, also quite interesting, in the inferior temporal cortex, which is also often named in the visual pathway. So I could use that too if I wanted to explore that further. The maxima are sorted by highest activity first and then lowest, but I can also now go back to my first static maximum and see dipoles from there for the initial primary cortical activation. So let me go there. Okay. Okay, and have a look at the maxima here. The first maximum should actually be right there, correct. So let me see another source here. And this is a very basic model that I've now created that explains my data actually quite nicely already. So I can invert this source and maybe move that up to the top. So I've got my initial cortex activation followed by the, this one should be inverted, followed by the empty activation. And I can also have a look at the Taylor coordinates for these sources and check whether they, which regions they belong to, for example. So yeah, that was just a quick illustration of how you could use the cortical imaging also to combine it with the dipole model. We could, of course, now refine this by, for example, fitting the orientation again over a larger time interval. Just really quickly before we move on about the settings. So this is for more professional users. You can go to the settings, and the settings for cortical Loretta and cortical Clara can be defined here. The most important ones are the SVD cutoff, which manipulates the regularization that is applied, the depth weighting on or off, and the Laplacian type, which is used. So by default, we use the unweighted graph, but there's also other ones, or you could even work without a Laplacian type if you wanted to. And then the number of iterations of Clara are defined here at the bottom. So currently it's set to 6 iterations. The more iterations you apply, the more focal the result will get. Okay, so this was the cortical imaging part. Now let's move to the age-appropriate template models. Just need to confirm that I'm not going to save the solution. And I would like to show you the age-appropriate template models with another data set. So this was a 12-year-old child that had epilepsy, and we had averages of several interictal spikes, and it's a combined MEG-EG recording. In this case, I would now just look at the EEG. So these are the 3 averages of 3 different spikes. The first spike has got the most averages, and we're going to work with that one. Just show you this in average reference. Right, and we're going to go through the same process again. So mark a block over the data interval, right-click, click source analysis, and define the interval that you really want to analyze. In this case, we're going to work with a symmetric interval around the spike peak from -200 to 200. And we've set the filters to a 3 Hertz forward filter for the high pass and a 35 zero phase for the for the low pass. So source analysis here. So we can see the data on the left, the global feed power, and let us simply start with by double-clicking into one of these head schemes here and generate a dipole model. So if I double-click here, I immediately set a dipole, and I get a model with a residual. Of course, I haven't fitted anything, so the residual is still very high. And I'm now going to use the PCA, click on the PCA button, to find a fit interval where I want to model the onset of activity. So we're most interested in the actual beginning of that spiking activation, and we're trying to find an interval where my PCA inside the interval explains more than 95% of the data. So this interval works quite well. We're going to start and fit this dipole, and we can now see that we've explained a lot of activity already, but there may still be a residual. And if I look at the residual over the whole interval, I can see there's still a large component that I haven't explained. So I will need a second dipole. So double-click again to set a new dipole. And now I can mark a fit interval over that activation here where my residual is highest and fit that second source over that residual. And I can now see that again, I have a better explanation of activity, but there's now still unexplained activity, and it's actually a rhythmic activity which seems to correlate with the spike onset. So I need another source for this, and since we're dealing with a rhythmic activity here, I'm going to work with a not a dipole source for this, but a so-called regional source, which summarizes the activity of a region in any direction. So I can click here to set a dipole and then use the C for convert key on the keyboard to convert it to a regional source, and now fit that regional source over the remaining time interval. So I'm going to work with a time interval, scale up these amplitudes, work with a time interval where the other sources don't have too much activation, and fit this regional source. Okay, and now I've got a fairly good explanation of the data. I've got 3 sources with separated waveforms, but of course I fitted these first 2 waveforms, these first 2 sources, not knowing about this rhythmic activation, which is actually even present when the other sources are alive. So let me just refit the whole thing over the whole time interval. So all fit, start fit these 3 sources all together. Yeah, and okay, I got a good data explanation, but I can also notice that this source here is extremely peripheral. This is actually the most important source of the onset, and it seems to be almost in the skull. And the reason for this is that I used a 4-shell ellipsoidal head model, and I really want to work with a model that is appropriate for the 12-year-old child that I'm working with. So I can now change my head model to account for this and work with the age-appropriate template model. So simply click into this model combo box and choose the age-appropriate template model, and get a warning that my source is no longer inside the brain volume, and I can choose in the next combo box that appears the right age group. So I'm working with a 12-year-old model. And now I want to do this fit again with this new head model. So all fit, start fit. And I can see immediately that the sources move way inside the brain. So the localization is actually totally different. I can check again using the undo function. So if I go to solution, undo last modification, or just press Ctrl Z, then I can go back to the previous. So this is the previous situation, and this is the one I get after the fit. Yeah, so obviously the head model made a big difference here. And let's just have a look at the anatomy. Press A for anatomy key. Right, and I can see that my source localizes in the left frontal cortex. Let's just compare this with the Clara results. So Clara is our volume imaging method. So if I just mark over the interval where my first source becomes active and choose Clara from this set of functions here, I can see how the Clara method runs, and the result, if I make this larger, is actually very consistent with my source position of my first source. So I've got a very good agreement of these 2 methods. Finally, I could also check on the cortical Clara again for this data set. So if I double click on the peak of the first source, so around about 10 milliseconds, I can choose the cortical Clara here. And this cortical Clara will now run on not the individual brain because we haven't co-registered it, but on the template brain for a 12-year-old child. Yeah, and in epilepsy, I'm often interested mainly in the onset activity, so the first spiking activity. Sometimes it propagates to a secondary location, which is also the case in this example. And This is also the reason why I took the forward filter for the high pass before sending the data up to source analysis, so that my initial activity is not confounded by anything that comes later, and possibly, if I use a zero phase shift filter, can bleed back and distort my activity of the first source. So now we are computing the cortical Clara. So it will run through the 6 iterations that we have chosen. And then, yeah, comes up with a result. So this is also consistent with the one I found from the dipole modeling. So it's in the left frontal lobe between a gyrus and a fissure here. And yeah, I could also use the inflation now to show this on the inflated. The inflated will look very similar to the one we've seen previously. And I think I'm going to skip this for time reasons now. And yeah, so this is just an insight into the application of age-appropriate template models, which we have also got available as a cortex reconstruction. And I'll now move to the last part of the introduction of the new features, which is about the default mode networks or other networks source montages that we offer. And to show you this feature, I'm going to work with another dataset again, which is about an error-related negativity experiment. It's also part of our example set. It's called correct plus error, and it contains 128 channels. They're already epoched, so this is why you see some jumps in the data. This is where one epoch ends and the next one starts. We can transform these into a source montage. So we could use the source button here and go to the resting state montages. And I'm now interested in the frontoparietal task control network. So there was a task here, a discrimination task, a visual task, and the subject had to press left or right mouse button depending on what the task, what the association was, and there was an error rate of about 30%. So we want to see if there's any difference between processing in error and correct, and what the network does. So first thing is I need to edit the paradigm. So open the paradigm file. We've already defined the conditions, so just to make things a bit quicker, we have 4 conditions. One is the stimulus followed by an error. One is the stimulus followed by a correct answer. You can see it's got more correct ones than errors. And there's also some 2 conditions that focus on the response, so triggered on the response given, but we're just now interested in the stimulus. So looking at the epochs, we made sure that the baseline is well before the stimulus, so there's not too much interaction between looking at anticipation. So one thing we need to do is perform an artifact scan to remove artifacts. The artifact scan gives us the trials in the columns and the channels in the rows. We can see a set of thresholds. We can change the threshold by moving this slider here to include or exclude more trials in the amplitude or in the gradient criterion. So basically we're looking at a threshold set that allows us to keep about 80% of the trials, which should be fine. Just wouldn't change too much here. Just move on to the coherence tab. And in this coherence tab, we can now identify 2 conditions, a target condition and also a control condition. So we're going to choose the stimulus as the correct as the target and the error as a control. And we work with all traces so that we don't lose out on any of the orientations that otherwise we would only use the radial orientation for each of these network sources. And we just want to make sure we catch everything. Right, and so this runs through the about 400 trials to compute the time frequency transform. I've chosen a resolution of frequency of 1 Hertz and time resolution of 50 milliseconds. That's just because I was more interested in the frequency resolution here than in the time resolution. So, and these are the different plots for the different sources, by default coming up in a temporal spectral evolution. So that means I see the percentual change to the baseline. And I can see in the in this RDIPFC source, which stands for right dorsolateral prefrontal cortex, in the radial as well as the vertical component at around 23 Hertz and then again 40 Hertz, there's some enhanced activation. Now in the standard correct, the stimulus followed by correct, and I can switch the task and see the similar and in the error condition. In the error condition, there's also some slow frequency, which we know is about the error-related negativity. Yeah, so this montage allows us to quite nicely pick out these activities and see if this network is involved in particular frequency oscillations here. I could also now look at coherences between for example, this frontal source and other, just by double-clicking on one of the source. And in this case, it doesn't seem to be too much coherence apart from other frontal sources. I could now it would make sense to do a statistical analysis if I had several subjects. So all these things can be exported and can also be batched together, so that I just need to set up the file, the paradigm, etc. once, and then I can run the batch quite nicely. Right, and yeah, so this leads me to the conclusion of this talk. If you want to learn more about the for example, how to batch data, how to export data for more analysis, etc. There's lots of information in our wiki, so wiki.besa.de, also of course on our website, besa.de, and yeah, the next webinar is about sort of this particular thing. So how do you put together lots of different subjects into statistical analysis using BESA statistics, which will be on Tuesday at the same time. And yeah, now this brings me to the end of the talk.